So our next speaker today is Theo Knijnenburg. And similar to Ilya, he started out as studying electrical engineering. And he got his master's degree at Delft University in the Netherlands. And this quickly turned to studying gene expression patterns and regulation, both in cancer and in yeast. And then Theo came as a postdoc at ISB in 2008. And his first paper with Ilya was entitled Fewer Permutations, More Accurate P-Values, which actually has a lot of citations. So please join me in welcoming Theo Knijnenburg.
Dave, thank you so much. My name is Theo Knijnenburg. It is a really big honor for me to present at this symposium. I'm very excited to start this afternoon session on cancer genomics with these three other fantastic speakers, Wei Zhang, Miyasha Chambwe, and Peter Laird. So, as Dave already alluded to, I am an electrical engineer by training, specialized in information theory, digital signal processing, machine learning, and statistics. I fell in love with this notion of being able to analyze, with computer technologies, biology, the stuff that life is made from. I worked at the ISB for a total of 9 years in the lab of Ilya Smulovich. And one of the main things that I worked on is cancer genomics. Specifically, this project, the Cancer Genome Atlas, TCGA, of which you have heard already quite a bit. You're going to hear much more about it in this session. So the TCGA is a very big project funded by the NCI and the NHGRI. We, which is hundreds of researchers in the TCGA, produced more than 2.5 petabytes of high-quality cancer genomics data. This is from tumor samples of more than 11,000 cancer patients who had one of 33 different tumor types. And so we interrogated these data using 7 different data platforms or modalities. This is gene mutations, DNA copy number variations, so amplifications and deletions, gene expression, epigenomic alterations in the form of DNA methylation, microRNA expression, protein levels from reverse phase protein arrays, and a lot of clinical data. I think TCGA was one of the foundational projects that gave rise to this notion of a real molecular characterization of cancer and cancer subtypes. I think we have already heard, and we will continue to hear more about this, that the TCGA has transformed and will continue to transform oncology practice. In the TCGA, we developed many domain-specific tools, such as very excellent mutation colors, to identify recurring mutations in oncogenes and tumor suppressor genes. And with this came, of course, lots of new insights, but also lots of new technology, right? And these tools that were developed, these algorithms, are now actually being used in a clinical setting. We ventured onto the cloud with our raw and processed data, and I think really pioneered the notion of secure and scalable cancer genomics data and compute in the cloud environment. TCGA is a very beautiful arena of research. I think many people's careers flourished within the TCGA, and many people that worked in the TCGA are now the new leaders in the area of cancer genomics. So for me, the key to the success of the TCGA is the ability to compare high quality multi-omics and clinical measurement data across many samples. Right? So indeed, you can envision your rectangular data table or your Excel spreadsheet, where on the rows we have all the TCGA samples or patients, and on the columns we have all this measurement data. And this allows us to do these statistical comparisons, visualizations, and also machine learning exercises. Of course, underlying all of this was a group of people with a vision to set it up and a large group of people that executed upon this vision. And so the TCGA was a US-based project which had many centers. And Ilya's lab here at ISB was a so-called genome data analysis center, together with the lab of Wei Zhang at the MD Anderson Cancer Institute. In the following talks, you will hear more about TCGA and about how this is transforming oncology practice. I want to mention one vignette here, which I think is cool for two reasons. One, the science and the application is really impactful. And the other is that my old roommate, when I did my PhD, is the senior author on this paper. He actually came to Ilya's lab in 2007 for an internship. I would like to think that he sort of paved the way for me to come to Seattle, to the ISB, which really set off my scientific research career. His name is Jeroen de Ridder. He's here on the right. We are visiting Ilya and Jeanette at their place in 2012. Okay, so I'm going to take you through this vignette, starting at the top left, all the way over there, and then in clockwise fashion, talk you through this story. So the title of the paper is called Ultra-Fast Deep Learned Central Nervous System Tumor Classification During Surgery. This is about brain and spinal cord tumors, which happen a lot in children and which are also one of the most lethal types of cancer in children. So there are many different subtypes of classes of CNS tumors based on the fact that they have different cells or locations of origin, but also the different oncogenetic or epigenetic processes that led to the formation of the tumor. Anytime that you want to operate on a tumor and remove it, you have to ask yourself the question, how much should you resect? Right? You want to maximally remove tumor tissue in order to prevent recurrence. And at the same time, you want to minimally remove healthy tissue. Especially in the brain, if you remove healthy tissue, this can lead to a dramatic decrease in the quality of life. With these brain tumors, it's very difficult, perhaps not even possible, to take a biopsy before you go into surgery. So they can do imaging, and they do imaging, but this does not usually lead to the good subtype classification. So when they do surgery, they go in and they don't really know yet what the subtype is. And the way in which this typically happens is by following the orange arrows, is that the pathologist gets a little bit of time in order to make a diagnosis, and in that way inform the surgeon of how much to resect. What they show in this paper is that they have a molecular diagnostics approach, genomic characterization based on DNA methylation, by which they are able to predict the subtype of the CNS tumor and help that to inform the actual decision in terms of how much to resect as part of this surgery. This is based on the notion that these various central nervous system tumors can be distinguished by their genome-wide DNA methylation pattern. So they have developed a deep learning model, which is called Sturgeon, which is trained on publicly available 450K methylation array data, which is the same methylation array platform that was used, I would say really spearheaded within the TCGA. And then they can do inference, predicting the subtype, using Oxford nanopore technology sequencing in real time. So this means as they start to sequence this sample and they get DNA methylation reads, they can already start to predict the tumor subtype. This is really the beauty of the approach from a machine learning standpoint. I will not talk about that. I will just show this picture, which is a timeline, where we can see that after, say, 15 minutes into the surgery, they already have the sample ready to be sequenced, and they start to sequence this sample, right? And then as time progresses, in this particular case, within 30 minutes, their algorithm is pretty certain that this subtype is a schwannoma. In the paper, they report accurate diagnostics for 45 out of the 50 retrospectively sequenced samples. For the 5 remaining ones, the algorithm wasn't wrong, it was simply uncertain, which is a very nice property to have in such an algorithm. In 25 real-time surgeries where they applied this, they get a correct diagnosis in 18% of the cases within 1.5 hours. So this notion of doing genomic characterization combined with machine learning during surgery in the operating room is a game changer for these types of tumors. Okay, so with all these advances, a question could be, are we able to cure cancer? No, is what I would say. I don't think that there's going to be a silver bullet for cancer. With the gift of multicellular life comes the curse of cancer. I had the opportunity to meet up with Sui a few weeks ago. We had a really great conversation. I would like to say that I started this sentence and that he finished it. Maybe this quote should be just attributed to Sui. At any rate, it's not just us humans that get cancer. Virtually all mammals can get cancer. Actually, most animals can get cancer. Cancer can arise in each tissue of our bodies. We identify circulating tumor cells in what seem to be healthy people. We know that the cells in our eyelids and moles have cancer-associated mutations. And from autopsies on males, it's clear that the very vast majority have the beginning of prostate cancer, this being completely unrelated to their cause of death. Another complicating factor in terms of curing cancer is that these cancer cells are our own cells. Right? So if we look at them genomically, which we did extensively in the TCGA, it's sort of a bioinformatician's dream, right? So many mutations, copy number variation. But from a therapeutical perspective, these cancer cells are very much like our own cells. Much easier to distinguish therapeutically between a cancer cell and a bacterial cell than between a healthy cell and a cancer cell. Many of the therapies today are pretty blunt instruments, one would say, right? So chemotherapy just kills dividing cells. We know that cancer cells are quickly dividing, but obviously it also kills and damages many other cells in our body. The final part of this argument is that cancer and evolution go hand in hand. Sorry, this is a bit faint here. I'm making the argument that cancer is everywhere, that cancer cells are very much like our own cells, and that cancer and evolution share mutation as a survival mechanism. The machinery to copy DNA and to repair DNA, they're very beautiful. It actually allows for mutations, which drives genetic variation, and ultimately over generations, adaptation and evolution. So this opinion that we will not completely eradicate cancer, of course, does not detract from the notion that we have made a huge amount of progress, and we will continue to do that in the prevention and in the detection and in the treatment and in the management of this disease. So what we will see is that we are able to save lives, to prevent the deaths of people that are still very much full of life and have a lot to contribute, like Ilya did. So I would like to switch gears, talk about Boolean networks, as many people have done already today. So in these Boolean networks, we have these nodes, which are either on or off. And these nodes are connected by Boolean logic functions. So Ilya, together with Ed Dougherty, Stu, and Wei, and others, spearheaded this notion of using dynamic Boolean networks to model gene regulation. In this particular case, a node is a gene, which is either on or off. And these genes cannot behave independently of each other. They are constrained by the topology and the functionality within the Boolean network. And the dynamics within the Boolean networks dictate that there are these stable attractor states that the system tends to move into. Back in 2009 or 2010, what I call the old ISB, the other side of the lake, Ilya once demonstrated how in the Boolean network you are able to move towards these stable states. And he did that by using the audience, or not using, as a Boolean network, where each audience member was a gene that was either on or off. So I've asked a few of you to actually come to the front a little bit so you know what is going to happen right now, which is, I think, a pretty big experiment. We are going to try and actually be a Boolean network. So everybody that is sitting here in the lower area, we're going to try and do this experiment together. And people that are sitting upstairs there, they are spectators in this. Okay. So I hope that you are excited to participate in this. Of course, also to make sure that you don't fall asleep after lunch. With this group, I would like you to, you have the chairs with the wheels to turn around a little bit to make sure that we can sort of face each other and are a group of sorts. And I'm going to ask each of you to identify 3 people. Those 3 people are going to be your input nodes. So the first thing that we need to do is we need to form a network topology. Are you participating in this, Lee? Okay. So yes, the only thing that you need to do is identify 3 people that are going to be your. Oh, no, not that it's, so we don't want all sorts of separate Boolean networks. We want one big Boolean network. So you're just going to identify 3 people sort of at random, but they have to be in your view such that you can see whether they are on or whether they're off. Yeah.
So what's on?
So let me first ask you this question. Does everybody in the lower area here see 3 people, which are their 3 input nodes? Yes? Okay. Remember those 3 people, they're going to be important in your lives for a couple of minutes at least. Okay, so this is the first step. The second step is that we have to decide on the Boolean logic that we are going to implement, which is going to be a majority voting, right? So we have, you're looking at 3 people, right? And this is a dynamic Boolean network, and we're going to step through this. And so this means that in the next update, it depends on the state of the 3 people that you're looking at. And once again, it is majority voting. So in the next step, you are going to lift your hand, be on, when at least 2 of your input nodes are on.
But you don't care which 2.
You don't care which 2. Majority voting. Yes. Very important.
Now, if you do it when with 2, that's just your little file. So you have to have everybody in.
The hope is that because people sort of randomly chose 3 persons, that if you look at the overall connectivity, we are one big network. So we can see if that is the case. You shouldn't change. So everybody still knows who their 3 people are that they're looking at. Okay, so we are with like, what is it, like 30 people here. So there's maybe a billion different initial configurations. And so we're going to choose one of those. So you're going to choose for yourself whether you are on or whether you are off. So please go ahead and choose whether you are on or whether you're off. And yeah, okay, once you make your decision, you have to keep doing that. Okay, excellent. Yeah, put it up high in the air, Bahar, please. Okay, so I'm gonna step. I'm gonna take this. I'm gonna go step. But first, I'm going to ask: does everybody know what their next move is going to be? If you look at the 3 people, you're going to do majority voting. Is everybody okay? If not, raise your hand. So please keep your hand up. Yeah. Yeah. Okay. Okay. So is your hand up or down, sir? Up. Okay. So we're going to step. So I'm going to say step, and then you're going to move to the next to the update based on the majority voting. Step. Okay. So now we're in a new state. Hopefully we're moving towards an attractor state. Does everybody know what their next step is going to be based on the majority voting? Yeah? Okay. So then we step. We step. We step. We step. We step. We step. I think everybody's on, right? Okay, great. Perfect. Maybe. So in a Boolean network with synchronous updates, there are two sort of stable attractors or types of stable attractors. One is a fixed state, fixed point attractor, and the other one is a cyclic attractor where you step through a small number of discrete states at the end. I think we reached a fixed point attractor. Okay. I propose that we do this once again. We keep the network topology the same. So you keep on looking at your same three people as an input. And you're once again going to decide for yourself whether you are on or off as an initial starting configuration. So please go ahead and decide whether you're on or off. Okay. And we're going to step again. And what I hope is that we're going to end in a different attractor state. This may work out or not. Everybody ready? Step. 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 Okay. Well, we did well, right? Obviously, when you have not 30 people, but 20,000 genes, you will see that these attractor states are that some of them are up, some of them are down, right? And the configuration of everything that is up may be indicative of the skin cell attractor state. And with the same gene regulatory network, you can get to brain cell attractor state, right? Okay, thank you. This was great, but not done yet. I'm going to try and do this once again, but now bring in the notion of cancer genomics and also how we understand gene regulatory networks, in the sense that some genes, if you will, are more important than others or are more connected than others. TP53 is certainly one of them. One way to look at this is a sort of big traffic light, which gets many different signals from the cell, and only when everything is looking okay, it's going to say, put the traffic light on green, and we can go into cell replication, so cell division. TP53 is one of the most frequently mutated oncogenes, which then means that the cell doesn't care anymore about the input that TP53 is getting, and TP53 is just going to say yes, go into cell division anyway. So I want to see whether we can also actually simulate that. So let us see. Boris, do you want to be my TP53? Okay, so this means that everybody is keeping their same three people as input nodes, but let's just say that everybody that's sitting behind Boris is going to discard one of their nodes and make sure that Boris is the third one. Is it okay for this group of people? So you have your three people in mind, including Boris over here? Okay.
Is Boris one of the three out?
For you and for a bunch of folks over here, not for everybody. But Boris is now a very densely connected node in the network. And what I hope to show is that he's more important than others. And we're going to do two more rounds. And what I will ask you is to remember your initial configuration. Boris is not mutated yet. We're going to step through that and end in some attractor state, which we're going to call the healthy state. And then my hope is that if we mutate Boris, where he's always going to be off, not listening to his inputs anymore, then we're going to end up in another attractor state, which we can call the cancer cell attractor state. Stu.
Technical question. Do the people who are looking at Boris give up one of their three?
Yes.
Or not?
Yes.
Randomly or
You can also choose new people if you want now. Yes, because we're still doing majority voting, so we want to keep it 3. That makes it easy. Okay, so I'm going to ask you once again to raise your hand and do an initial starting configuration. Okay. And so nothing has really changed right now other than that many people have Boris as their input node. Okay. So let's step, 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 step. I think is everybody on? Yeah, okay, great. So I guess my hope now is that if we turn Boris off, TP53, but we start with the same configuration as we did before, then we're going to end in a different state, which may be with this group of people that everything is going to turn off again, which would be nice in this particular case. So please go back to your initial configuration of just now. And Boris, of the third try that we played, yeah. And Boris, you don't have to look at anyone right now. You will just keep your arms down. You are mutated, yes. And we're gonna step, 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 step. Okay, great. So the only thing that we changed, we did not change the network topology. The only thing that we changed is that we have a cancer gene, which no longer cares about what other genes in the network do. It's just always off. And you see that this pushed the system into a different attractor. Once again, perhaps the attractors were not super attractive because everything was on or everything was off. But I hope that sort of this demonstration shows a little bit how in a dynamic way you can actually move to these attractor states within this Boolean network context. So I thank you very much for participating in this. I'm going to continue with the presentation. You can relax. I'm running out of time here a little bit. So this notion of using dynamical networks to model gene regulation is not far sought at all. Here we're looking at hematopoiesis, where you see how from the pluripotent hematopoietic stem cell, we can differentiate into all sorts of blood types, including the red blood types and the white blood cells, which are your immune cells. At this stage of the common myeloid progenitor, over here, there's this interplay between two genes, two transcription factors, GATA1 and PU1, which are repressing each other, and they are activating themselves, leading to this dynamic attractor landscape where we have three states: an unstable state, C, which is like a marble on top of a hill, and then we have two stable attractor states, A and B, which are the marbles in the valleys. If these cells get pushed to A, where GATA1 is high and PU1 is low, they will follow the erythroid lineage. They can, for example, become red blood cells. If they end up in B, where PU1 is high and GATA1 is low, they will move towards this myelomonocytic lineage. They can become neutrophils or macrophages. The dynamic Boolean networks is an instance of a dynamical system where small particles interact with each other to produce these emergent state-like behavior. I would argue that we see that everywhere in the world. Take water. like any other substance that can exist, if you will, in three phases: solid, liquid, gas, three states. Water is water. You start raising the temperature, it's still water, until you push it over a particular critical boundary. Then it undergoes this state transition and becomes steam. Although our lives are sort of obviously continuous, at the same time, we go through various states, being awake and being asleep. When we look at a butterfly, we can certainly identify very clear states. What also these dynamical systems seem to do is that if these particles have simple rules, they can still lead to complex, intelligent-like behavior. So I would say that it is very intuitive to go and use dynamical systems to model gene regulatory networks. And if you can do this well and actually have a good mathematical description, you can now start to understand the system, and you can start to come up with strategies in which you're able to control the system, push the system toward a desired state, away from a disease state towards a healthy state. Big challenge in gene regulatory network inference, and we will hear about this in the presentation by Wei Zhang after this one, is finding a good impedance match between the measurement reality of your data and the mathematical model that you're using to model that. I would say at this point in time, we often default to defining states as clusters in a high-dimensional space. We can measure so many things from one data point or from one sample that we then do a dimensionality reduction, a UMAP or a t-SNE, and there are our points, and we draw a circle around it and say this is the state. This is perhaps circumstantial evidence that underlying this is a dynamical system and that this is a state, but just drawing a circle around it obviously does not give us the control that we ultimately want. In the last few minutes, I'm going to very briefly talk about the work that I'm currently doing. I'm working at the Altius Institute for Biomedical Sciences, which, like the ISB, is a nonprofit interdisciplinary research organization. We are in the waterfront area. It's a 25-minute walk to ISB. Our mission is to advance the science of gene regulation for the benefit of human health. Altius has been a big player in the ENCODE consortium, which investigates the non-coding regulatory part of the genome. We could say that we are here in 2024, quite a while after the Human Genome Project. We know a lot, and at the same time, we know little, right? In the sense that the function of most human genes remains undefined, or at least ill-defined. Current studies to figure out human gene function and regulation often apply knockouts. They are done in cell lines, and they are often statistically underpowered. We are asking the question, what if we can turn the dial on gene regulation in primary human cells? I'm rushing through all of this. We are using a molecular technology, similar perhaps to CRISPR, which is called transcription factor activator-like effectors, TALEs, which allow us to point to a particular place on the genome. We send the protein there. We're not cutting into the DNA, but we let it bind. And together with epi-effector domains, CREB for downregulating and VP64 for upregulating, we can actually tune target gene expression. So what we are looking at over here is the expression of the gene CD96 measured by flow cytometry. We're trying to repress that gene with our TALEs. And we can see that we have a range of different repressors that are going from very effective to a little bit less effective. We are doing an experiment which we call internally the MegaMap, where we are taking primary T cells, which we individually activate or repress. So we don't do this in a pooled fashion. We use an array-based fashion, where each gene perturbation is in an individual well. We split these cells, we measure the downstream consequences using both single-cell RNA-seq as well as the single-cell imaging. And we want to do this for thousands of cells per perturbation. At this point in time, we are trying to activate and repress all the genes that are expressed in these T cells, which is on the order of 10,000. We are at this point in time in a production phase where we are generating a million sequencing data on 1 million cells per day and imaging data on 10 million cells per day. This is for me a very fast pace, but really great interdisciplinary environment with a very strong interaction between the wet lab and the computational lab in order to make this happen. Okay, with this, I am rounding up. Sorry for going over time a little bit. I would like to say that Ilya's mentorship, his kindness, and his wisdom have been life-changing for me and also career defining. I miss him a lot. Vladimir, Marina, also Jeanette, and Anna, thank you so much for being here today. Ilya has been a really great gift, not just to me, but I think to everyone. It's a gift that we can only use for good and for love.